Hello, and welcome to the lecture on Chapter 7, the continuation of the lectures on Chapter 7. We're going to continue with microbial genetics this week. The last bit that we had covered on the previous week, we had DNA replication. That was the copying of the DNA that had to happen before the cell could reproduce and give genetic information to the two daughter cells. This week, we're going to be focusing on the transfer of information from DNA, and like really what is the DNA for is providing information so that we can build proteins. And I say we, either eukaryotic cells or bacterial cells do the same process. So let's have a look. We're going to talk about the fact that genes, they're sections of the DNA that code for proteins. And all of the genes of an organism put together, that's what we refer to as the genotype. And then there's a related concept, which is which of those genes are being transcribed and translated to be made and give the physical appearance or characteristics of an organism. That's the phenotype. And I'll remind you, not every gene is transcribed in every cell, and not every gene is transcribed simultaneously. So it's the particular combination of genes that are being transcribed. I'll remind you that those two terms, the transcription and translation, these are the two main steps of the so-called central dogma of genetics. We have DNA information, that's the code for making proteins, but we can't go directly from those instructions straight to making proteins. We have to go through this intermediary molecule called RNA. And it's not that the DNA is becoming RNA, it's the information in the DNA, the sequence of those letters, A's, T's, G's, and C's, that gets copied using RNA molecules. So you're making a version of this information that then can be used for the next step, which is translation, which is taking that information of messenger RNA and using that as the template to bring the correct amino acids in order to make a long strand of amino acids. The bonds between the amino acids are called peptide bonds. So that long strand is referred to as a polypeptide. It's getting synthesized according to the instructions in the RNA. Again, it's not that DNA becomes RNA or that RNA becomes protein. It's that the instructions in those different molecules are used to build the next molecule. The cartoon version of this, here's the double-stranded DNA. Just like when we copied the DNA, before we can transcribe the DNA, we need to pull those two strands apart from each other. And then another difference from copying the DNA that both sides would be copied. When transcribing the DNA, only one of the two parts of the DNA, one of the two sides of the DNA is actually being transcribed. In case you are one of those folks who get these two terms mixed up with each other, most people can remember it goes DNA, RNA, protein. And so the step from DNA to RNA, that's transcription. And the step from RNA to protein, that's translation. These are in alphabetical order. Transcription would come before translation in the alphabet. C comes before L. Okay. One more thing about this. When we do that second step, that translation step, the messenger RNA is read 3 letters at a time. You remember codons, the AUG, it's the universal start codon. It always means bring me a methionine, this amino acid. The next one, CGG, means bring me an arginine, and so on. And remember that table, it's like 64 different combinations. Those different combinations, that's a universal code. So in us eukaryotic cells and in bacterial cells and in viruses for that matter, this code is the same. It's going to mean bring me this particular amino acid, and this means bring me this other amino acid. I always like to point out how universal that is in all the species. All right, so a little video on transcription.
Cells use DNA to store information while proteins are used to perform the activities necessary to keep the cell alive. In a process called transcription, the information stored in a DNA molecule is copied into RNA molecules, which can be used to synthesize specific proteins.
Okay, I'm going to stop this. What is happening here is transcription. So what's represented in brown, this is an enzyme called RNA polymerase or RNA polymerase. The ACE tells you it's an enzyme, and RNA polymer tells you what it's making. The double-stranded DNA has to be pulled apart, and then only one of the strands gets transcribed. This RNA polymerase is making messenger RNA, mRNA, for us. And then, as we'll see, for the next step, for the translation step, we need two more kinds of RNA. We need a ribosomal RNA, that is an organelle called a ribosome that joins with the messenger RNA, that joins with the red molecule here, and allows another helper molecule, these transfer RNAs, to come in and read one codon and bring the correct amino acid, and then the next codon gets read, and so on. And that is how we do the next step, which will be in another video. Let's see, we're almost done with this one.
Cells use three types of RNA: ribosomal RNA molecules, which are used to form ribosomes; transfer RNA molecules, which deliver amino acids to ribosomes; and messenger RNA molecules, which carry the information for making specific proteins from DNA to ribosomes.
Okay, so that's the messenger RNA. That's what's being represented here in red. Now let's go on to translation. This is a piece of messenger RNA here with a whole bunch of different ribosomes on it doing this translation step. In other words, all of these things that look like kind of chains of pearls, all of those are amino acid chains that are being built off of the instructions as one set of instructions, and you're making a whole lot of proteins off of this. It's the same protein. You're making the same chain of amino acids in the same order to make the same protein.
Translation is the process by which the information contained in a molecule of messenger RNA is used to synthesize a protein. The process
So again, we have the ribosomes. There's a small ribosomal subunit and a large ribosomal subunit. They have to come together. And that's similar to eukaryotic cells. They have that as well. What's a little bit different in its bacterial ribosomal subunits are a little bit different. And so actually that makes a really good target for drugs that combat bacterial infections without harming our eukaryotic cells. Let's see. So the two pieces of the ribosome come together. They start reading this messenger RNA. We need transfer RNA molecules coming in to bring the correct amino acids. And then this chain starts being built and gets longer and longer and longer. As you can see, these proteins are all being made the same way. And then eventually get to the end of the instructions, and the two ribosomal subunits come apart, and this strand of amino acids gets released, and then it has to get folded.
This is called translation because ribosomes read along a messenger RNA molecule, translating the genetic code into the amino acid sequence of the protein. Because there are no nuclei in bacteria, translation can actually begin before transcription is completed.
So that's different from eukaryotic cells. Remember, in eukaryotic cells, the DNA is stuck in the nucleus, and so the transcription step happens, and it's isolated in the nucleus, and that messenger RNA has to leave the nucleus to find a ribosome to read it and translate it. But in the bacterial cell, there is no separate nucleus. There's just that DNA molecule, that round DNA molecule. And so transcription can happen. You start making messenger RNA, and right away, the ribosomes are right there. So if the ribosome connects with the messenger RNA as the messenger RNA is being made, then the ribosome can start working on this. I picture it this way, that it's, I'm reading the book's the DNA, that's the information, I'm transcribing, I'm writing the notes, but then somebody behind me is looking over my shoulder and using those instructions to build whatever it is. These are the instructions to build. So those two things are kind of happening as we work. I transcribe, somebody else translates, and we're doing it at the same time. Things that have to happen in transcription. We have to get it started. So there's RNA primers have to be made, little strips of RNA. Remember when we talked about the beginning of DNA replication, copying the DNA, we also had to make a primer, like a little strip of RNA in the beginning to get things started. Then the long strand of information, that's the messenger RNA. We've already talked about ribosomal RNAs and transfer RNAs, regulatory RNAs and ribozymes. I'm not going to go into in huge detail. What's happening in the nucleoid of prokaryotes. Remember, nucleoid means that that's where the DNA is. You have to kind of get started, initiation of transcription. Then we continue elongating, making the longer messenger RNA strand, and then finally you got to stop. That's termination. And here's a video covering those.
There are 3 stages in the process of transcription: initiation, elongation, and termination. Let's see how each stage works. During initiation, RNA polymerase binds to the DNA and recognizes a site called a promoter at the 3' end of the template strand of the target gene.
Okay. I know that that's a lot of terminology. The 3' end just means this is the end where you can start. This is like an on-ramp. This is where we can get started. So initiation is the coming together of this RNA polymerase and the correct section of the DNA.
When RNA polymerase finds a promoter, it breaks the hydrogen bonds holding the DNA strands together at the site of the promoter, and transcription begins.
And then it's going to continue.
RNA polymerase does not bind to all promoters with equal affinity. This difference in promoter strength is one way that cells can control gene expression. The more strongly RNA polymerase binds to a particular promoter, the more likely that gene is to be transcribed.
Okay, so the promoters, in some sense, are better. The promoters for genes that need to be transcribed a lot, the ones that we need a lot of copies of this protein, those are going to have, in some sense, more attractive or stickier promoters. Like, start here, please. We need more of this particular gene.
In general, higher levels of transcription lead to higher levels of translation, which leads to higher concentrations of that polypeptide. One strand of the DNA serves as a template strand. The RNA transcript is copied from this strand of the gene and therefore has an RNA sequence complementary to this strand.
Okay, so in case you were like me, as a student, I used to always be just obsessed with this, like, well, which strand is it, that gets transcribed? It doesn't matter. Well, let me rephrase that. So a particular gene might occur on the upper strand of this DNA molecule, but a different gene might occur on the bottom strand of the DNA molecule. Makes no difference because you've got the complementary base pairing. So for a particular gene, you've got the kind of the money side, the one that's actually has the information, and then you've got kind of the dummy side, the side that's just complementary to it, and it's there kind of for ease of copying the DNA. But when it comes to transcription, only one of those sides of the gene is going to be the information side.
During elongation, RNA polymerase moves along the DNA, opening up a bubble in the DNA. As it moves, RNA polymerase adds bases to the 3' end of the growing RNA transcript.
So just getting longer. And then finally, we're going to wrap it up. to end.
Elongation continues until RNA polymerase reaches a site on the DNA called a terminator. At this site, RNA polymerase and the newly synthesized RNA transcript are released from the DNA.
I think of this as kind of the end of the roller coaster ride. You know, like you get to the end and then everybody disembarks, gets off of this ship.
Termination can occur through two mechanisms, self-termination and enzyme-dependent termination. In self-termination, the RNA sequence transcribed at the terminator causes the RNA to hydrogen bond with itself, forming a stem loop structure which essentially pulls the RNA polymerase off the DNA. In enzyme-dependent termination, a termination protein binds to the terminator and pushes RNA polymerase off the DNA.
So that's just details about two different ways of ending this ride. In some cases, it's this very last bit of messenger RNA is kind of sticky to itself, and the molecule is likely to fold over onto itself, and that's how it disengages from the rest of these structures. In the other case, there's actually a physical thing, a protein that's standing here, and as this DNA, as this RNA polymerase arrives, it's kind of pushed off the rails. Either way, it comes to an end, and this messenger RNA molecule is released. So the whole process, again, in cartoon, the double-stranded DNA gets pulled apart. There's a section of it called a promoter that makes it easier for this RNA polymerase molecule to arrive. Sigma factors they make it even easier for this to happen. I don't know what to say other than like it's like calling over the RNA polymerase. Come here, buddy. Like, let's get started. It makes this more likely to happen. The gene then gets transcribed. So we start doing the kind of sort of copying, except we're not making more DNA, we're making RNA instead. So that's represented here in red, this initiation. So we start making this RNA primer, and then we just keep moving on into elongation. And you can have multiple RNA polymerases happening, or you can think of this as happening through time, that this RNA polymerase keeps moving down, and as it moves down, this strand of messenger RNA is getting longer and longer until finally we reach the terminator, and then we're done transcribing. And finally, termination can happen because we get to a part where nearing the end of this messenger RNA, and when it circles back onto itself and starts sticking to itself, that's not going to be able to stay in contact with the DNA, and so this thing's going to fall off. Or you can get to a termination protein. Something's going to like bump this thing off of the rails. So to summarize, in transcriptional differences in eukaryotes. So how this is different from what's going on in the bacteria are a few things. One, we already mentioned, RNA transcription has to happen in the nucleus of eukaryotic cells, not like prokaryotic cells. Because there's also some DNA, those plasmids and mitochondria and chloroplasts and those organelles, there's actually some transcription happening in those sections of the cell also. We have the three nuclear RNA, it doesn't matter, a bunch of transcription factors, things that are going to make it happen more readily. And then here's one difference with eukaryotic RNA processing. There's some additional steps before we get to the translation step. There's a cap put on the molecule. There's a bunch of A's, adenines put at the tail, poly-A tail, polyadenylation. And what that's about is, look, I'm about to send this messenger RNA out into the cytoplasm, outside of the nucleus. I need it to connect with the ribosomes, but I don't need it to get chewed up right away and destroyed. And so that's what the capping and polyadenylation are. And then splicing is one thing that eukaryotic cells do that prokaryotic cells don't do, and it's basically editing. So here's that in a cartoon. This represents a strand of messenger RNA. And if we were talking about a prokaryote, that's it. That's what you got, the messenger RNA, and that's got to go get translated. But with eukaryotes, there's all this editing. There's these pieces that were added in that were referred to as introns. So that stuff gets cut out to indicate that it's different from the introns that are stuck in. The name for the other pieces that get left behind and used, they're referred to as exons. Doesn't matter. The point is, you get all the exons, put them together, put a cap on one end and a poly A tail on the other end, and that is what gets to leave the nucleus and go get translated. Okay, and that is the end of part 3. This first part for this week, but 3 overall. And I will see you over in part 4.